Hello, this is Mr. Arlt, and I'm going to go over quarter two test review. So this is on cell topics and other overviews from this quarter. This is actually aligned with the worksheet where it's question and answer as well. And so this is just about those topics. These are the main things you need to know for the test. So here's some of the stuff we're going to go over. Again, it's just review. It's nothing new. It's just in case you want to get a one size fits all kind of overview. I'm going to talk about microscopes, talk about plant and animal cells with organelles. We'll go over diffusion, osmosis again, talk a little bit about mitosis, and then photosynthesis and cellular respiration. So microscopes, we did a little bit with microscopes. We didn't go over all of the parts, so don't stress about that. The big things that we want to make sure that you remember is that a microscope reverses and flips the image on the slide. So if we're looking at the slide, it would be right under here. And so the ocular lens is this eyepiece. So that's where that would be. And the objective lens are the ones that we often change. And they go from 410, maybe 40, 100, depends on the microscope. And they each have a magnification actually. So what we do to get total magnification is just multiply them together. So ocular lenses, here's an example for us. Ocular lenses tend to be 10x, so 10 times magnification. So it's not super high, but those objectives can be anything from 4 to 100, pretty standard. So in this example, if we were looking at the yellow one, which is actually not on, that would be 40x, and we multiply those two things together, and we get 400. And we write that as 400x, 400 times magnification for the total magnification. We viewed three main types of cells under magnification. So we looked at onion root tip cells, cheek cells, and bacterial capsules. To identify these, there's a few things we could look for. As we were learning about the different types of cells, we know that some have cell walls, some don't. So in the onion root tip cells, if we were trying to figure out, is that onion root tip? Well, we'd look to see, is it sort of squarish or rectangular? You can see these hard lines, and those are cell walls. That's a really good indication you're looking at a plant in general, which an onion is. They have these dark purple centers, and that's actually the nuclei, the nucleus. And they contain genetic material in them. And so we can actually, that purple portion is actually the genetic material that's been stained. Then sometimes we can see organelles, little bits and bobs kind of floating around within the cell wall boundary. And we definitely can see cytoplasm. We can see a section around the nucleus. In the cheek cell, some of those are the same. We can see nuclei. We can sort of see that purple genetic material. We may be able to see organelles. So you can see a bunch of dots in here. My guess is that that's ribosomes or some other material in there. And we see cytoplasm. We don't see cell walls. They're blobby, they're round. And so between these two, we can tell the organization of this onion root tip one looks very grid-like. And we don't get that with the cheek cells. They're independent, they're kind of amorphous, they don't really have a structure. The bacterial capsules are very different. This particular picture isn't the one from our site. However, we can't really see anything. They're so small, we really just can't tell anything except the outside of the capsule. You can kind of see this area around it. We can't really see anything in it. We can't see any nucleus. They're super tiny. And so if we see little things that are just shapes, rods, spheres, spirals, but we can't tell anything else, it's probably a bacteria. So we can tell the difference between plant and animal cells. And remember that plants and animals are all eukaryotes. So we actually need to know the difference between a eukaryote and a prokaryote. So we're just looking at this, if I go back here, between a bacterial capsule versus either the onion or the cheek cell. So the prokaryotes are going to be much smaller. We just saw that. You could tell, you can easily see it under a microscope. However, if we're just looking at one of these diagrams and we can't really tell size, then we look inside. You're not going to see these membrane-bound organelles. Okay, there's not a lot going on. You just got some ribosomes and a few other things. You do have cytoplasm, but there's no nucleus. You've got DNA kind of swimming around in here in this other configuration that's different than our cells. And then if we were looking for bigger things, they reproduce. Binary fusion is one of the big ways that they reproduce. There are a few other ways we learned about, but in general, binary fusion. Eukaryotes, however, have a whole bunch of stuff going on inside of them. They have all these different organelles we'll talk about later. They're far bigger. We can actually see many of them with just minor amounts of magnification, 10 times magnification. They have a very clear nucleus. And then these go through mitosis and then meiosis. We did not talk about meiosis this quarter, but it is one of the other cellular processes that they use for reproduction. So let's take a look at some of the organelles that we highlighted. Now, many of the pictures you're going to see in this look a little bit different than, say, our diagram. This is a slice through version of a cell, and so we get a different view of some of them. What we want to know in what I'm going to go over is actually the functions. So I'm going to show you real images. So many of these are photographs of microscopes under a microscope. So the cell wall we talked about, that's this rigid border, this thick border around the outside, and that provides structural support for the cell and a nice barrier. Now, this is only in plants. This is not in plants and animals. We don't see this in any animal cells. The cell membrane, so now in this picture, if I look just on the inside, I can see another line going along the inside. It's slightly lighter. Now, that's the cell membrane. Animal and plant cells both have cell membranes. These are semi-permeable. We call them phospholipid bilayers. And that's because we have these phospholipids, and there's two of them. Bi means two, layers, layer, so two layered of phospholipids. And we got this little head on one part and these little fat tails on the other part. And this allows certain molecules to move in and some to move either in or out of the cell, actually. But some things can't, and they require these channels and other things. And we'll talk about this a little bit later as well. The nucleus is this purplish part, and that's just a part containing the DNA within any cell that has them in eukaryotic cells. Vacuole. This is largely something that we see in plant cells. We do see them in animal cells, but they're much smaller. They play a very different function of just minor storage of materials and molecules. In a plant, they really provide a lot of support for the cell. That's and increase the amount of pressure that the cell can have so that with some water loss, it can actually balance out some of those things. The ribosome, these are just little dots. Now, this ribosome picture that we're seeing here is actually also rough ER. However, I'm going to go back a little bit so you can see in here, ribosomes can be free-floating as well. And so they can be throughout the cell. And we see these in prokaryotes as well as eukaryotes. But these things are assembling amino acids into proteins for the cell. So they're making a whole bunch of things. These are our builders. We find them on the rough ER, and these are just these little foldy things. I'm actually going to go back a little so you can see they're right next to the nucleus. And so they're taking instructions from the nucleus and turning them into actual materials. So they're located near the nucleus, and these build them, but then also on the rough ER, they're checking that protein, make sure everything's good. The smooth ER takes those proteins and then preps it for transport. And it preps it for transport within the cell. So the Golgi apparatus, which we see is floating away from the nucleus, actually takes materials and transports them out of the cell. We've got two ways we can make energy. We have the mitochondrion, which we find in both plants and animals, and that takes sugars and turns it into usable energy. And then we have chloroplasts, which also make energy, but it's storing energy from sunlight in the form of sugar molecules. And then we have an organelle called the lysosome, and this is breaking down wastes within the cell. So they're the cleaner upper, the erasers, the vacuums, however we want to take it. They take molecules, envelop them in their little balloons, and then they have different chemicals and enzymes that can break those molecules down into component parts that the cell uses then. So it's digesting all of these molecules. So organelles are performing specific cellular functions. And though animal and plants share a lot of them, we do have some differences. So just as a reminder, we don't see the cell wall that we see in this picture here. We really don't see a significant vacuole. Again, there are some animal cells that do have this. Largely, those are protista, not our cells like ours. Though I don't want to tell you it's never, but it's not worth remembering it as an animal function. And then we don't see chloroplasts really in anything other than plants. They are the significant chloroplast holders. So let's just take a look at this diagram version of the cell so that we can see them as well. They look a little different often in the diagram. So we've got our cell membrane, the outside layer. A nucleus usually looks like a round blob in the center. We've got ribosomes. So if we see little specks floating throughout the cell, those are our ribosomes. We've got the rough ER, and that's going to be next to the nucleus with little dots on it. That's why it looks rough. And then the smooth ER does not have those ribosomes on the surface, and that will also be near the nucleus. Golgi apparatus looks very similar to the smooth and rough ER. They often have these little blobby bits. It kind of looks like a lava lamp, and it's not next to the nucleus though. Mitochondria are little jelly beans floating around with little folds on the inside. We've got lysosomes. They're going to look like little bubbles. They're hard to sometimes distinguish. And then we've got centrioles as well. So depending on the stage that the cell is in, we may or may not easily see centrioles around or doing anything. These are important for mitosis. In the plant one, they look very similar. We just have a few extra bits. So we've got a cell wall, a thick layer on the outside, as well as a cell membrane, that same phospholipid bilayer. We still see a nucleus. We see a large vacuole. Often it's the bulk of the center of these plant cells. We see ribosomes, our little specks floating around. They're also on the rough ER and near the smooth ER, which are both by the nucleus. We see our Golgi apparatus still. But now we have a chloroplast, a green jelly bean, as well as our mitochondria, the other kind of jelly, the more bean-shaped jelly bean. And then we might also see lysosomes floating around as well. Diffusion is one of the ways that we get things in and out and around in a cell. And that's the movement of a molecule, also called a solute, within a solvent. So a solute is something, a salt or a sugar or a molecule that's added and can dissolve within a solution, which by going into a solvent. So for instance, we can dissolve salt within water. Salt is our solute. The solvent is the water. The molecules that we put in, so in this case, if it's just red dye or pink dye, if we watch it over time, just general action will spread those dye molecules pretty evenly throughout the solution, and that's equilibrium. And this is really, we want to think about this as concentration. So they go to a balanced concentration over time. So in our own bodies, diffusion, this is often how oxygen gets into, or in this case, out of a cell. It's going from high to low concentration. So if there's a lot of oxygen within the cell, it's going to force the oxygen to move outside if there's low oxygen levels outside of the cell. Now, this is a passive form of movement, so there's no energy that's being used at all. These molecules just move kind of on their own. However, if we increase the temperature inside of this, in this picture, if we were to have hot water, it would actually happen faster than if it was cold water. It would still happen, it would just happen faster. Osmosis is Very similar, except we're talking about water moving. Now, as you can see in this diagram, in this little animation, there's a semi-permeable membrane. That means that there's only some things, semi, that can actually pass through this membrane. So in this case, it's just water. The other molecule that's in here is actually too large. It's a sugar molecule, so it's too big to fit through the holes in that membrane. It's still going to try to seek an equilibrium, a balanced concentration of the solute. And the way to do that is to actually pass water molecules to the other side. It's just too highly concentrated, and so it's going to move over. This results in actually an increase in the amount of water on one side versus the other. So this is how water enters like a red blood cell. So if we have a lot of sugar inside of our blood, it's going to change the way that blood is, the water is moving throughout the cell. This is also passive, so the water is moving on its own. There's no additional energy required for it to move. But water pressure can, if we increase the pressure on one of the sides, we can actually force it to go in a single direction. But in general, within our own cells, there's no extra energy required. So here's a quick little comparison Venn diagram. So both of them happen over time. They both move from a high concentration to a low concentration and move towards equilibrium. In diffusion, we're actually talking about the solutes moving. We don't necessarily have a membrane involved. Though if we're talking about things diffusing across cell boundaries, then there is a membrane, the semi-permeable membrane of our phospholipid bilayer. The solvent level stays the same. So the water, for instance, isn't going to change inside one of them necessarily. And temperature will largely impact the speed of of diffusion. In osmosis, we're talking about the solvents moving. So we're talking about the liquid part, the water. And then this does need to happen across a semi-permeable membrane. So we are specifically saying water is moving into or out of a cell. The solvent level changes, right? So the water level inside one of those situations is going to change. So the cell is either going to have more water or less water. And pressure is the thing that really impacts this. So let's kind of swap over to mitosis. So mitosis, once a cell reaches a certain size, it needs to change. It needs to divide. Diffusion slows down and can only works very efficiently in cells of generally a specific size, a smaller size. When we're too big, it just takes too long for things to get through, and the cell actually loses out. So it's good for cells to divide. This is called the cell cycle when we look at it from growth, the period of growth to division to growth back to division. Okay, so we call that the cell cycle. Most of the time that the cell is growing, we call interphase. So you can see here, there are actually two big G phases, which are growth phases, and then an S phase, which is when the DNA replicates. During mitosis, we result in two daughter cells. So that same cell gets big, 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 preps for mitosis, divides, makes an exact or relatively exact copy. The DNA amount does not change. Okay, there's 46 chromosomes in the original cell and 46 when it's done. We call these diploid. Okay, so we start with a diploid cell, we end with a diploid cell. Okay. There are 4 stages that we want you to remember. There's prophase. This animation is not synced with where I am. We'll see it here in a second. I'll sync it when we're there. We've got prophase, metaphase, anaphase, and telophase. So we can see the end part of telophase right now, and cytokinesis happening. So as it starts here, I'll kind of narrate through. Okay, so we've got 2 daughter cells, and now G1, G2, and S, all those things. So now we're in interphase. So we've got prophase here. It's ready to divide. So we can see the DNA condensed, and the DNA, the nuclear membrane, the spot around the nucleus, is actually dissolving. As we go into metaphase, those chromosomes lined up. We have all these spindle fibers that attach to them, and now in anaphase, they're going to separate away from each other so that we end up with two nuclei. And then in telophase, those spindle fibers dissolve, the nuclear membrane reforms, and those two cell nuclei are ready. And then something called cytokinesis happens, and that's pinching the cellular membranes apart into two cells. Okay, so we end up with two daughter cells that are more or less exact copies of each other. So this regulates how cells change over time. This is important. We have ways in our body to say that this is going well, this is not going well. So the cell cycle is normal. This is how we need to replace old cells. So healthy cells continue dividing normally, okay? Sometimes, though, there's something that happens and a cell is damaged. So if we can't repair it, the body performs apoptosis, is what it's called. And so the cell just sort of dies off and goes away. And that's great, so that we don't pass on something that could be harmful to the body. Now, in cancer, we have unregulated growth. So the cell cycle just kind of keeps on happening and happening and happening. So we call that uncontrolled cell division. And this is why we get things like tumors or other growths within the body. So cancer is a very diverse set of things, and many things can cause it. But in general, it's just uncontrolled cell division. So the body no longer can regulate how often and to what extent a cell grows. So usually there's cues that happen from neighbors. There's not enough room for another cell, so it'll wait. It's big enough for now. Or all the cells are happy and healthy, so we don't need to keep growing. With cancer, this doesn't occur. So this is a mitosis-based issue, though. Now, all cells have to use energy and kind of get energy. The two main molecules that we talk about in biology for energy are ADP and ATP. They're really more or less the same molecule. They just have one small difference, and that's a phosphate group. So ADP has two little phosphates at the end. That's these P molecules here on this tail. ADP is an empty battery. Still very important because ADP is rechargeable. It's a rechargeable battery. If we add a little energy, plug on the phosphate here, we get a third in the chain, and we end up with ATP. Now this is adenosine diphosphate up top. That's two. Di is another form of like biphosphate, diphosphate. And ATP is adenosine triphosphate, so three phosphates. This is a full battery. This is a charged up molecule. There's energy stored in the bond between the second and third phosphates. And we can extract that later on to run cellular functions. And that's really, really important. So this whole main process of ADP to ATP, this is basically the main thing that our mitochondria are doing for us. That's why we call them our powerhouse. So they're powering us up. They're filling up all the batteries for us. So let's take a look at photosynthesis though first, because photosynthesis is where we get the stored energy that actually gives us our full batteries later on. So we have reactants and products here, and our reactants for photosynthesis are carbon dioxide and water. And then those use sunlight to store energy as sugar, and then it releases oxygen as a second product. This is happening in the chloroplast. So there's a couple of things that we should see. There's an inner and outer membrane. There's two membranes. And then we've got these stack of coins called thylakoids. And those are all floating around in stroma, which is the fluid that's inside of this little M&M jelly bean thing that's the chloroplast. So there's two steps. there's one step that requires light, and then there's another step that doesn't require light. So the non-light one is happening pretty much all the time, day and night. However, the first step here can only happen during daylight, unless we have a grow light to synthesize additional light. It requires light of some kind. So these are called light-dependent reactions, and water that's in the cell is taken into the chloroplast, and it's broken apart, and it forms ATP and NADPH molecules. And then it ends up releasing oxygen because we're stripping the hydrogen of the water. So we use that hydrogen to charge up ATP and then also charge up this NADPH. So these are just energy batteries that the cell uses. And then in the Calvin cycle here, the light independent reactions, that energy that we charged up, those batteries, are used to be taken carbon dioxide, and then it goes through a series of steps to convert it into glucose or sugar. And that's the product we want. That sugar gets stored and used. So it can be, there's lots of different things that are used. Plants store sugar as parts of the tree mass, all sorts of things. And then all of that so all of this is happening within the chloroplast. And that's important because it's slightly different than what we see in cellular respiration. Now, cellular respiration is taking those sugars and converting them to ATP. Now, plants can they have chloroplasts, most of them. They can make their own sugars and then use those sugars to make their own ATP. Animals and other consumers have to then consume either a plant or another animal to get those sugars. We can't produce our own off the bat just from the sun. So we have to, for instance, consume sugar, breathe in oxygen, and then we can run a series of reactions in our mitochondria to produce carbon dioxide, water, and then a bunch of ATP. So we react sugar and oxygen together and produce carbon dioxide, water, and energy. Now, this is all happening in the mitochondria more or less. You're going to see there's one step that actually happens in just the cell. Those mitochondria look very similar. We've got an inner and outer membrane. There's a space between that's really important, between that inner and outer membrane. And then we call the inside of the mitochondria the matrix. These folds are the cristae, and there is DNA that's inside. This is in both plants and animals, though, okay? So everybody's doing this part. Only plants do photosynthesis, but everybody does. Plants and animals are doing cellular respiration. So there's 3 steps. You got glycolysis. This happens outside of the mitochondria. It happens before we get in there. So the main, within just the regular part of the cell, in the cytosol, that's the fluid part, the cell takes oxygen and breaks down that sugar into a form called pyruvate. Now we make a few additional side things. So we make an NADH battery, and then we make a couple ATP, but not a whole lot. So we're just getting a little bit here, but we need to break it into that pyruvate. So now that pyruvate is shipped into the mitochondria, and then step 2 takes over. So inside the main matrix of the mitochondria, we perform the Krebs cycle. And then what that Krebs cycle does is it just breaks that pyruvate down into more NADH, and FADH2 batteries, like a lot of them, a good chunk of them. So we charge up a whole bunch of these batteries, and we make a couple ATP, just a couple. And then the true magic happens. And so in step 3, all of these charged up NADH and FADH2 batteries are in this inner membrane space right along this little purple line here, purple and mauve and pinky, whatever color. We're going to see the electron transport chain take over. And what it does is it is able to actually just make a bunch of ATP. So we recharge ADPs into ATPs. So we take all these byproducts that we made in these earlier steps and funnel hydrogen ions through ATP synthase, a special protein, and it makes all these ATPs. And so that's 34 that we make in this one process. That's a lot. Water and carbon dioxide are byproducts of this. So that's why we end up having to expel CO2. It's part of this process of cellular respiration. So if we look at the comparison here, we can see that photosynthesis has CO2 and H2O as reactants, glucose and oxygen as products, and it uses energy from the sun to store them as sugar, to store energy as sugar. And then in cellular respiration, it's the exact opposite. We take glucose and oxygen to make carbon dioxide and water. We're using stored energy, so we don't actually require really any energy, a little bit to get things started, but not much. And then we release a ton of ATP, about 36 total. That if it's not completely efficient, it can be a little bit less, or a little bit more if it's extra efficient. And then this happens in animals and plants, where only really plants are doing photosynthesis. There are some bacteria and some other organisms that can do that as well, and not every plant can, but more or less everybody can. These are connected to each other, so they're just flips. And so I really like this diagram because I think that you can really see the connection. So both systems are putting oxygen and CO2 out. There's water that's being cycled back and forth, and then sugar out on one side, sugar in on the other. And ATP is the name of the game. We're trying to charge those batteries. So this is, again, just a review. So look back over, go back through, return to these. You can turn the sound off and kind of skip on through if you need to look at another slide. This is just to help us as we get ready for Q2 test review in just about a week or so. So happy reviewing, and I hope this was helpful, and we'll talk to you all later.